So in this Unit 3 video for Advanced Higher Bio, we're going to be looking at types of study that can be done. So the first type of study that we're going to look at are animal studies. And often in biology, animal studies are unavoidable. But where animal studies are used, it does often lead to advances in medicine. And in situations where the use of animals is unavoidable, there are three concepts: replacement, reduction, and refinement, in order to reduce or minimize the harm that is done to animals. So replacement, what that means is substituting an animal for something else. So can a cellular model, a 3D printed version of something be used instead of animals? The second one is reduction. Now this often looks at the experiment and in a pilot study uses the results to predict what is the minimum number of organisms that need to be used in order to get statistical results, and making sure that you use the minimum number of subjects rather than the maximum number of subjects. The third one is refinement. Now refinement is a difficult one to explain, but refinement is looking for ways which would minimize the suffering to the animal welfare and human error. So refining your technique so that you're not causing undue pain to organisms. In some situations, organisms could be modified so that they don't experience types of pain, or where it is necessary to see a pain response, that the time threshold that you're allowed animals to experience pain for is kept to a minimum. Now, human studies are ones involving humans. And when it comes to humans, the most important thing is consent. And consent is not just saying, yes, I agree to this. The subjects need to be made aware of everything that's involved in the process. And that becomes informed consent. A healthcare provider might ask a patient to consent to receive therapy before providing it, or a clinical researcher may ask a research participant before enrolling that person into a clinical trial. If participants are under 16, you must get consent from a parent or carer, as anyone under the age of 16 is not considered legally able to provide informed. Also within human trials, there is the right to withdraw. So participants must be aware that at any point they can withdraw from the study and therefore withdraw all their data from the investigation. And researchers must inform subjects that the right to refuse to participate or withdraw their study at any stage for whatever reason. If, however, the investigation is closed, you lose your right to withdraw after a certain period. It is only alongside consent is the notion of confidentiality. Participant confidentiality must be maintained. Confidentiality pertains to the treatment of information. So all the information that a person has revealed about themselves and had the expectation that would not be shared with other people, that must not be shared with other people. Now, the justification for scientific research must also assess the risks. And only when the potential benefits outweigh the risk can you say, or the mere pursuit of scientific knowledge, is it justifiable in doing the work. By providing informed consent, participants are assessing for themselves that they consider the work to be of more benefit than the risk. The scientific investigations are not purely scientific in nature. There are factors that influence the type of studies that get conducted and the type of studies that are done more frequently. So legislation, regulation, policy, and funding all participate. So there is a legal requirement for certain risk assessments to be done that would take into account the safety of humans and animals involved and any impact on the environment. If regulations would be infringed by that research does not go ahead. The regulation, policy and licensing of governments which aim to limit the potential for misuse of studies and data, limit how much data you can collect, what is considered informed consent, etc. And finally, funding will influence the direction and pace of scientific progress. We've seen this when it comes to science related to climate change. The science was conducted and was known for a very long time that there was a man-made correlation between the impacts of humans and climate change, but money prevented that from really being widespread, and it was only private companies that themselves were highly polluting that had access to that research. Additionally, if you are a professor working in a university, you get funding in order to hire your employees. Whichever company or agency gives you money will expect you to do research that suits their sensibilities, and there are certain types of research that cannot be done. For example, in America, it is prohibited to investigate any of the effects of guns on society. You're not allowed to research it because it's seen to infringe upon the right to bear arms. Also in America, it is not possible to do any research into the medical benefits of marijuana because of its legal classification in American law systems. So there are certain things that prevent research from being done. When it comes to studies, we can have in vivo and in vitro studies. And the first one we'll look at is in vitro. In vitro means in glass, and it is when we look at biological molecules or cells in a very controlled environment, so they're not in their normal biological system. An in vitro experiment is very easy to control all the variables, but is very limited in scope. It will not reveal any potential effects that would be noticed in other parts of an organism or in any systems of an organism. Usually, in vitro would be the first type of research or investigation that is done to determine cellular effects before you would move on to trialing in animals. And that would be seen as part of the refinement process, making sure that there is an effect before you would even think about putting it into an animal and potentially causing. In vivo experiments are where you test your pharmaceutical or your experimentation on a whole living organism. The benefit of this is that you get to investigate the whole overall effect on all parts of an organism and its systems. In vivo testing of animals must be performed by law before you can advance to human clinical trials. Now, when it comes to drugs, the advantages of in vitro work is that it allows you to control all the variables, these confounding variables, which we'll discuss in another video. The disadvantage of in vivo is that when you're working with patients or you're working with animals, they may not remember to take the drug or administer it correctly. So you've got a loss of control, which will impact on the effectiveness. An advantage of in vivo is that you do get to see the effect on the whole organism. And this is a very frequently asked question. What is the benefit of doing in vivo work? You see the effect on the whole organism. Now, when you're doing in vivo and you use a control would often be a placebo. A placebo is a drug that is usually something like sugar that will not have a biological consequence, but it allows you to compare the effect of the drug to not. Another set of experiments we have are laboratory experiments versus field studies. A laboratory experiment is one carried out in an experiment under very controlled circumstances. It's very artificial. But because it's in a laboratory, it tends to be very straightforward to do. However, not whenever you're housing organisms or humans. It's very difficult to do laboratory experiments on people. A field experiment is when you're doing the research where the organism gets to be within its natural setting or habitat. These experiments are standard for psychology and environmental biology, where you're not going to be trapping things and getting them to exist in laboratory conditions. The benefit of field experiments is that they will provide results which are more applicable to a wider biological setting. You get to see the effect of whatever you're doing, not only on the organism, but on how it affects how that organism also interacts with other things. But the biggest drawback is the lack of control and how much you do not know. A clinical trial is when you investigate a medicine's impact on humans, and it is by nature a field experiment because the people are living in their own homes. And it would be very impractical and also very costly to try and have humans live in a controlled environment for an extended period of time. Clinical trials allow researchers to investigate the performance of a medicine while people are going about their daily routine. And it may reveal that there are certain activities, certain foods, certain drinks that might have an impact on the effectiveness of a type of medication. And as said before, the drawback is difficulty in controlling all the variables. As part of a clinical trial, patients may be asked not to drink. You cannot control whether or not they drink or not if they are in their own homes. They may be requested to do a minimum amount of exercise. That cannot be controlled. So there is a lot of unknown. Field studies also apply to plants. Whenever new plants are bred, it's usually to improve their hardiness, so how well they survive, or how much yield they produce. Often in laboratory conditions, you will see noticeable effects, but the overall growth of a plant is not just due to its genetics, it's also due to environmental factors. And so it is really essential after a new plant species or cultivar is developed under laboratory conditions that they get tested in a field environment. Finally, we have observational studies. Observational studies is when we look at a group that already exists rather than a group that gets instigated as part of a body of research. It's very easy to identify that observational studies are done in ecology, where you're looking at populations of wild animals. But in human studies, an observational study would be such as looking at the relationship between smoking and lung cancer. It would not be ethical for a researcher to get a group of people to start smoking if they suspected that smoking causes lung disease. Instead, they will be looking at only people who already smoke, which means there is bias because there may be factors, biological factors that cause someone either to start smoking or be addicted to smoking that might also be factors that lead to lung disease. So in this study, obviously, if you're looking at lung disease in smokers, the only group you've got are smokers. By recruiting experimental groups this way from a group that already exists, it will allow you the benefit of looking at the long-term effects of a particular behaviour. Because if you were trying to do that from scratch, it would require you to get someone to do something for 20 years. And if it started to have negative effects on their lives, they are likely to stop. Now, because observational studies are based on groups that already exist, there is a lot that cannot be controlled because the group has already existed and been doing a certain activity for a while before the researcher gets involved. Which means there is a large potential for bias, which may affect the results of the study, and so there is no truly independent variable, and a lot of it is based on qualitative research rather than quantitative research. However, observational studies themselves are good at detecting correlation, but less good for causation because so much work has already been done prior to the groups themselves being recruited. So that's been our video on types of experiment.